faithfully delivering value, interpreting great books from around the world. Hello, this is Sally's Book Talk. Today we're exploring the book Apprenticeship to Genius, The Making of a Scientific Dynasty. American science writer Robert Kennegal interviews scientists and publishes their stories in the New York Times Magazine, the New York Times Book Review, and the Wilson Quarterly. Now, when Robert wanted to write about Johns Hopkins neuropharmacologist Snyder, who'd gained international fame by age 40, his hard-won interview went surprisingly poorly. Snyder gave only canned responses straight from magazine profiles, so Robert deliberately mentioned Snyder's mentor to shift the dynamic.
Oh, Snyder lit up immediately, passionately recalling his work with the mentor 20 years prior. That pivot saved the interview and revealed a grander story spanning 3 generations of scientists.
Encouraged by his editor, Robert interviewed pharmacologists from NIH and Johns Hopkins, ultimately writing Apprenticeship to Genius. The book reveals how, in America, a scientist's early career depends half on whose lab they trained in, half on their discoveries. And data shows over half of U.S. Nobel laureates were once trainees of other Nobel winners. Apprenticeship chronicles 5 legendary biomedical scientists, including Snyder, in a multi-generational mentor chain, showcasing mentorship's profound impact. Its 14 chapters chronologically trace these pharmacologists' stories from the 1940s to 1990s along this mentorship lineage. I'll break this down into 3 parts. First, let's meet these 5 scientists and their extraordinary achievements. Second, what invaluable scientific philosophies were passed down this mentor chain? Third, the contradictions and drawbacks in the mentor-student inheritance culture. First, let's meet the 5 main scientists covered in the book: Shannon, Brody, Axelrod, Rhodes, Snyder, and Pert. These scientists are giants in pharmacology and neuroscience. 3 received Nobel Prize nominations and 1 won the award. Pharmacology focuses on studying how drugs work, providing the basis for developing new drug uses and pharmaceuticals. Pharmacology uses experimental methods. Scientists at packed lab benches constantly seek new ideas to test how drugs metabolize in the body, adjusting doses or discovering useful new compounds. Experimental facts. Shannon is the origin of this mentor-student lineage. He's a legendary figure, widely recognized for groundbreaking research in kidney function, chemotherapy, and malaria. When summarizing Shannon's scientific feats, it's said he almost single-handedly turned renal physiology from observational to highly precise quantitative science. Shannon designed precise experiments showing kidney glomeruli filter 180 liters of blood daily, with 1% forming urine. Before this, no one could answer that question. However, the author highlights Shannon's most praised trait: his leadership in spotting talent and motivating research teams. The story starts in World War II, when U.S. troops in the South Pacific suffered from malaria, but quinine couldn't be supplied due to Japan's occupation of the Dutch East Indies. So in 1942, Shannon was tasked with a special wartime mission at Goldwater Memorial Hospital, researching anti-malarial drugs. Shannon had a keen instinct for excellent research talent. His former colleagues estimated that nearly all 15 key researchers he recruited during this period later held pivotal roles in medicine. For instance, as an institute director or university department head, Shannon was calm, serious, and highly logical. Almost everyone who worked for him admired and revered him. The antimalarial research was successfully completed in 1946. Afterward, Shannon left Goldwater Hospital to become director of the Squibb Institute for Medical Research. There, he helped confirm the efficacy of a new antibiotic, streptomycin, and pushed for its mass production. However, because the Squibb Institute was too commercially driven, in 1949, Shannon left Squibb to establish the National Heart Institute for the government under the NIH. Many dismissed it as bureaucratic and doubted his research capabilities. Yet through his influence, Shannon recruited brilliant young scientists, transforming the Heart Institute into one of the world's most productive research units. Thus, in 1952, Shannon became NIH Deputy Director, rising to Director by 1955. The second link in this mentor chain is Bernard Brodie. Brodie was born in Britain, came to America for grad school, and became a pharmacology research assistant at NYU School of Medicine under Shannon. And their labs shared a building. Shannon recognized Brodie as promising, not just skilled but unconventional. He often had groundbreaking ideas that challenged scientific norms. So when Shannon took over a new lab at Goldwater Hospital in 1941, he offered Brodie a position. Brodie gladly accepted, entering his golden research era. Brodie deeply respected Shannon, almost viewing him as a father figure. He inherited Shannon's drug measurement approach, refining it into his own innovative methods.
In 1947, Brodie published 6 consecutive papers on false integration assay techniques in the Journal of Biological Chemistry, revolutionizing pharmacology. How can you take the king?
So what is the false integration technique? Before, scientists could only guess drug effectiveness from patient reactions. This technique let them precisely measure drug concentrations throughout the body to optimize dosage. Thanks to this breakthrough, from the late 1950s through the 1960s, Brodie earned international honors like the Torald Salmon Pharmacology Award, Lasker Award, and National Medal of Science. Moreover, he was elected to the Czechoslovak Academy of Sciences and U.S. National Academy of Sciences and received an honorary doctorate from the University of Barcelona. So, Brodie's scientific reputation grew ever higher. His fame spread worldwide, and his most famous disciple was Julius Axelrod, the third scientist we'll introduce. Axelrod was quiet, humble, and kind. Before meeting Brodie, he was unknown, working at a nonprofit lab in New York testing vitamins. One day in 1946, his supervisor posed a puzzle. Why did some headache medications worsen headaches and cause dizziness or diarrhea? This was beyond his expertise, so his supervisor sent him to seek help from one person, Brodie. They met at Goldwater Memorial Hospital, a meeting Axelrod would later recall as life-changing. Years later, Axelrod described this as the day that determined his destiny. They discussed for 3 hours. Brodie's insights were so profound they later co-published a paper on their findings. From then on, Axelrod embarked on a research path under Brodie's mentorship. Together, they studied drug metabolism pathways, revolutionizing painkillers, anticoagulants, caffeine-based drugs, and more. When Axelrod later led his own lab, he discovered norepinephrine could be reabsorbed and stored after release.
This discovery rewrote textbooks.
Norepinephrine, a key neurotransmitter, facilitates neuron communication and affects attention, mood, and sleep. For this achievement, Axelrod won the 1970 Nobel Prize. Crucially, enzymes identified in this research enabled treatments for hypertension and schizophrenia, leading to new antidepressants. Axelrod made extraordinary contributions to pharmacology. The fourth scientist is Solomon Snyder. Snyder was Axelrod's most renowned protege, exceptionally bright and ambitious from childhood. After graduating from Georgetown Medical School, Snyder went to Kaiser Hospital in San Francisco as an intern. A year later, family reasons brought him back to Washington, just as a position opened in Axelrod's lab. Snyder secured the position by leveraging his strong reputation in the field, beating out numerous competitors. He officially joined Axelrod's lab in 1963. Snyder and Axelrod jointly pioneered research on histamine. Histamine is a hormone released when body tissues are injured or inflamed. Their technique could detect histamine at 1/1000th of a gram, enabling inflammation detection throughout the body. Snyder was exceptionally prolific. Most scientists consider 100 lifetime papers an achievement, yet he'd published 400 by age 40. His landmark discoveries include nitric oxide as a neurotransmitter and the dopamine hypothesis for schizophrenia. Johns Hopkins named its experimental science department after him. His awards include the Wolf Prize in Medicine, Bauer Award, and Ralph W. Gerard Prize. The fifth scientist, Candace Pert, initially majored in English literature but loved biology. When applying for grad school, her husband's military posting in Maryland led Pert to target Johns Hopkins. Through connections, she submitted credentials to Snyder. Snyder was then Hopkins' youngest full professor. After gaining his approval, Pert formally applied. In 1970, she joined Snyder's lab. Snyder had many bright students, but none matched Pert's caliber. Snyder and Pert together made groundbreaking discoveries on how drugs affect the brain. Well, this work cemented Pert's status in pharmacology and neuroscience. At merely 26, she had reached heights beyond those of many scientists who'd struggled for decades. By 1984, Pert had published 110 papers and won the American Chemical Society's Arthur Fleming Award. She gave numerous lectures and served on editorial boards for 5 scientific journals. Having met these 5 scientists in the mentor lineage, what makes this scientific family unique? And how did they achieve such remarkable scientific success?
Now let's explore part 2. 3 invaluable scientific philosophies passed down among these top scientists.
First principle, study important problems at the right time. It sounds simple, but execution's tricky. How do you judge timing and a topic's significance? You won't find answers in books. While many researchers lingered on ill-timed or irrelevant topics, Brodie to Pert lived this principle, achieving breakthrough after breakthrough. But pray, through daily collaboration with mentors, these pharmacologists developed crucial intuition and judgment, passing it down generations. Consider a perfect pace of right timing meeting right topic.
When Brodie joined Shannon's lab as a research assistant, he hadn't yet made his mark.
His imagination and unconventional thinking led Shannon to assign him a sub-problem in anti-malarial drug research. Malaria was World War II's foremost medical challenge. The anti-malarial drug Atabrine had severe side effects, preventing effective dosing. Brodie's task, measuring Atabrine blood concentration, was notoriously difficult. He devised an indirect approach, measuring Atabrine metabolites post-digestion. Ultimately, Brodie's team invented a groundbreaking method to measure concentrations in blood, urine, feces, and tissues, applicable to humans and animals. Animal tests then revealed most injected Atabrine accumulated in muscles or livers with minimal blood presence. This explained why U.S. troops saw no effects. Dosage couldn't sustain blood concentration. Based on this, the team redesigned dosage protocols, solving the anti-malarial crisis. Brodie played a pivotal role. This project launched his career. His reputation soared, forever shaped by the project's urgent, agile wartime environment. For the rest of his career, any research project that wasn't thrilling simply didn't catch his eye. He passed this high standard for research topics to his own students. The second perfect timing meets topic example involves Snyder and Pert.
Pharmacology long assumed drugs worked by binding to receptors in organisms.
But in the 20th century, receptors remained theoretical, discussed but unproven. As drug abuse surged among U.S. troops and white youth, receptor research became urgent. Addictive drugs like heroin and morphine were called opioids. Scientists believed opioids acted on something in the brain, opioid receptors. Proving these receptors existed and understanding their mechanism could solve addiction. Considering its societal impact, positive potential, and likely government funding, in early 1972, Snyder entered this field with student Pert. Their key tool was radioactive naloxone, an opioid antagonist that theoretically occupied opioid receptors. Snyder and Pert injected opioids and naloxone into mouse brains, then filtered, cooled, and washed the tissue. They then measured brain radioactivity levels. Pert discovered that adding opioids reduced radioactivity in filtered mouse brains. Higher opioid concentrations caused bigger drops.
This proved opioids competed for brain receptors, with higher concentrations having greater effects.
For comparison, Pert tested non-opioid drugs against naloxone. None competed for receptors.
Thus, Snyder and Pert confirmed opioid receptors' existence, finding especially dense concentrations in the brain's limbic system.
Their paper, published in Science in March 1973, opened new doors in receptor research. Many scientists dedicated years to studying opioid receptors with no success. But Snyder simply seized the moment when the U.S. government prioritized drug issues, making it his research focus. Using extraordinary ingenuity, he and Pert proved the receptor's existence within a year. Overnight, Snyder and Pert became scientific superstars, especially Snyder, whose lab gained fame, attracting grad students and resources. The second philosophy, passed down, boldly experiment to find answers. Brody's catchphrase was, "Let's give it a bold try." Scientists in this lineage acted immediately on worthy problems. No endless prep, no paper reviews, just trusting intuition and doing. Start with a simple experiment, just try it. Axelrod first met Brody to consult about painkillers. Acetanilide was the active ingredient, but it caused dizziness and fatigue. Brody hypothesized, since headache and fatigue are classic methemoglobinemia symptoms, acetanilide might produce it, but how? Simple. Run a quick test right away. First, check acetanilide levels in patients' urine and feces after taking it. Axelrod found no acetanilide at all, meaning it transformed into something else. But what? Brody guessed aniline? It's just acetanilide minus an acetyl group. They gave aniline to dogs. Even Axelrod took 50 milligrams himself, and methaglobin levels spiked. This evidence confirmed Brody's hypothesis. Exactly. Acetanilide's toxic side effects came from aniline, so what chemical actually relieved pain? Using similar methods, they detected acetaminophen in patients, confirming its pain relief without methemoglobin risks. Axelrod and Brody's scientific discoveries led to the widely used fever reductor and painkiller today, Tylenol. In their subsequent collaborations, they also studied the metabolic processes of many drugs. Axelrod fully inherited Brody's bold approach of conducting simple experiments and passed it on unreservedly to Snyder. So much so that even decades after leaving Axelrod to start his own lab, Snyder would repeatedly recite Axelrod's famous saying, "An experiment is only worth doing if it's easy to do." When facing a new problem, compared to an experiment that requires meticulous preparation, high complexity, and waiting days or months, he would rather spend 3 hours doing a few rough experiments to roughly gauge whether a problem is worth researching. And Snyder's apprentice Pert also embraced this philosophy. Others described her as very aggressive, bold, always quickly shifting, constantly seeking new directions to advance research. The third, continuously passed down important philosophy is to unleash unlimited imagination. Before developing a tabrine, Brody had studied the efficacy of a South American bark against malaria. This bark contained 4 alkaloids. 2 were quickly detected, but the other 2 couldn't be traced in the blood. At that time, gas chromatography hadn't been invented, and these 2 alkaloids had no fluorescence to track. What to do? Brody spent 4 consecutive days at the New York Public Library, searching for inspiration, reading all kinds of literature, even papers on German dyes. It was this literature that gave Brody a brilliant idea: to dye those 2 alkaloids and use their color intensity in solution to detect concentration. Alkaloids are alkaline and combine with acidic dyes to form salts. If lucky, the salt would carry the dye's color.
Brody immediately called to order various acidic dyes, bringing back hundreds of types.
He and his colleagues tried them one by one, working continuously for 32 hours. In the end, only 2 or 3 salt particles had color, but unfortunately, it faded quickly, making this approach seem unworkable. Just as Brody, full of disappointment, was preparing to go home, he spotted methyl orange on the lab bench. Methyl orange is a commonly used reagent in laboratories that changes color to reflect acidity changes in solutions.
Brody suddenly thought to try it. Problem solved in 5 minutes.
He invented methyl orange assay. Mix blood extracted drugs with methyl orange to form salts, then measure concentration via calibrated colorimeter. This fusion of dye chemistry and pharmacology achieved a technological breakthrough. It convinced Brody that, with imagination and hard work, he could measure any substance. He relentlessly passed this philosophy to students by posing imaginative questions and encouraging wide reading. And, for example, if we weren't biologists but chemists, or nature itself, How would we design this biological mechanism? Axelrod, Snyder, and Pert naturally inherited this style. Their minds roamed freely across disciplines, drawing inspiration from diverse fields. The scientific community widely recognizes mentorship's role in cultivating talent, passing down elite thinking and research capabilities across generations. So is this mentorship flawless? Absolutely not. Next, we explore conflicts and drawbacks along this mentor chain. The first conflict arose between Brody and Axelrod. Their first 4 years were harmonious. Axelrod was talented, quickly becoming Brody's star technician. But as Axelrod gained experience, he longed to create his own projects and conduct independent research. So,
Sadly, Brody's lab offered no such opportunities.
moreover, Axelrod lacked a PhD for legitimate independent work, and Brody discouraged him from pursuing one. Clearly, Brody didn't want to lose Axelrod, creating growing tension. In 1952, with Brody's reluctant approval, Axelrod began nearly independent research on ephedrine and amphetamines. These stimulant drugs, addictive and commonly called narcotics, after figuring out how these two drugs metabolized, he began searching for enzymes that aid drug metabolism. Enzymes are biological catalysts. Many biochemical processes can't happen without them. With the scientist's friend's help, Axelrod proved how microsomal enzymes to catalyze multiple drugs. Traditionally, enzymes were thought to act only on one or a few specific substances. This discovery shattered conventional wisdom and greatly advanced drug processing technology. When Axelrod published his research summary in a conference journal, Brody recognized its potential. He gathered the whole lab to expand this research project. Then they'd publish a joint paper in a top-tier journal, listing everyone as co-authors. The joint paper caused a sensation, but Axelrod's name was completely overshadowed.
Feeling Brody had stolen his work, Axelrod resolved to leave.
For years after the incident, Axelrod refused to speak about Brody. The scientific community widely discussed this event. Indeed, credit disputes and mentors clinging to star apprentices are common in mentor-student relationships. The second example is Snyder and Pert's conflict, more acrimonious, even affecting Snyder's Nobel chances. Earlier, Pert became famous for co-discovering opioid receptors with Snyder. She was once grateful, but by 1978, everything changed when the Lasker Award went solely to Snyder. The Lasker Medical Award is America's most prestigious biomedical prize, second only to the Nobel. Lasker recipients often later win the Nobel Prize. Thus, the medical community calls it the Nobel Predictor. For the opioid receptor discovery, the Lasker was awarded to Snyder but not Pert. Learning this, Pert was furious. She urged Snyder to reject the award in protest or share prize money with her alma mater. But Snyder refused both, leaving Pert devastated.
She felt Snyder didn't support her recognition.
So she wrote to Mary Lasker, the award founder's widow, calling the decision unfair. Excerpts were published in Science. Soon after, Science news reporters covered this, raising gender discrimination views and causing a huge stir in the scientific community. Pert's letter's impact is still unfolding. The next year, the announcement of Nobel Prize winners was postponed, and the Physiology or Medicine Prize went to the developers of the CAT scanner. Snyder, who was expected to win, lost out. Insiders mocked that this year's Physiology or Medicine Prize became an engineering prize. Clearly, Snyder was eliminated at the last minute. As everyone knows, the Nobel Prize dislikes scandals. The Swedes have a simple belief: focus on research and avoid scandals. Snyder and Pert's relationship broke over this, and similar disputes happen a lot in science. This shows it's sometimes tough to pin down who made a scientific discovery, especially when genius teachers and top students collaborate. Besides credit disputes and mentors not letting go, are there other downsides to mentorship? In this star-studded mentorship lineage from the book, such drawbacks might not be obvious, but it really happens. If you can't get into such a mentorship, it's hard to make your mark. Despite these flaws, no denying mentorship helps science screen top talent and cultivate it. Young scientists' ability to judge topics, their research style, and how they'll mentor their own students later. The great scientist Newton long ago revealed mentorship's secret. He said, "If I have seen further, it is by standing on the shoulders of giants." If great scientific progress can't be done by one genius alone, it needs countless scientific families passing the baton through generations to push human knowledge's boundaries. All right, Apprenticeship to Genius. That wraps up our interpretation of this book. Reading notes are below the audio. See you next time.